Boa tarde a todos. É com imenso prazer que dou o início ao nosso segundo dia com os nossos palestrantes internacionais. É um prazer para mim poder estar aqui representando, o nosso programa de pós-graduação de Ciência Animal e a Universidade Estadual de Londrina. Então, muito obrigado à presença de todos, ontem e hoje, e nós iremos dar início, então, à nossa primeira palestra de hoje. Então, agora eu vou virar a chavinha e a gente vai começar a falar em inglês. So, for today, we will have Dr. Jussara do Carmo. Dr. Jussara is Associate Professor in the University of Mississippi Medical Center. Dr. Jussara started her career in UMMC in 2006 as a postdoc. After that, she had the positions of instructor, assistant professor, and in 2018 started as assistant professor. Dr. Jussara's research has focused on transgenerational obesity and understanding how the central nervous system and the leptin melanocortin system regulate metabolic and cardiovascular functions. And today, Dr. Jussara will talk to us about intergenerational obesity and its impact on kidney and cardiac function. Dr. Jussara, thank you so much for your time with us. Thank you so much for preparing this speech to us. We are very, very happy with you here. And as I always say here, for me, you are one of example of the power of the woman in science with rich disposition in USA that I really appreciated to have my time with you. So thank you so much for sharing your knowledge with us. Thank you, Jussara. I wish you a good speech.
Oh, thank you, Fabio. And now you almost make me cry. First of all, I would like to thank you and the whole committee for the invitation to talk a little bit about our research. That has been a good journey for us. Since I moved here, I started to work with obesity, and I have been learning so much, not only professionally, but as a person. I think it's a very, very good experience to be abroad and see a lot of things that happen here that could be happening in Brazil. And I'm so glad that you, as a young investigator, started a program and you are leading this kind of talk that is very, very nice, and sharing with other institutions in other departments. It's very, very, very nice from you guys.
Okay, thank you.
The developmental origin of health in disease concept has highlighted how the fetal and postnatal environment is shaping the long-term health. And the topic of my talk today will be to show the long-term consequence of parental obesity on the developmental program of metabolic and cardiorenal disease in offspring. Obesity has rapidly become one of the most important health problems worldwide. As you can see here in this slide, we have rates of obesity that range from less than 10% up to 40%. And just looking at the South America here, for example, you can see that Brazil, in most of the countries in the South America, we have a rate of obesity range from 10 to 20%. Interesting that Venezuela, as you can see here in dark, the obesity rate there is around 30 to 40%. That's very similar what's going on here in the United States, part of the Middle East, as you can see here. And in some countries, we are almost getting what we are facing here in the United States. For example, you can see here in the Australian that have the 20 to 30% of rate of obesity, as well Russia and Canada. And obesity has increased dramatically in all of the countries, especially here in the United States, as I mentioned before. And this prevalence has increased in the past 2 decades. This increase has occurred in children, adolescents, and adults of both sexes in all ethnic groups. We are here located in Mississippi. As you can see here, our prevalence of obesity is around 39.5%. And it's not, I'm not glad to tell you that we are the #1 in the country in rate of obesity. And as you can see, the hit when you have more obese, we have the increasing prevalence of hypertension and also diabetes. In fact, obesity is a major independent risk factor for hypertension and also diabetes and metabolic disease and other metabolic disease. In fact, obesity, hypertension, and diabetes are leading cause of chronic kidney disease in its progression to end stage renal disease. The pathological mechanism of chronic kidney disease are complex and include mechanical stretch of glomeruli, podocyte injury, oxidative stress, endoplasmic reticulum stress that we call ER stress, mitochondrial dysfunction, and apoptosis. However, the mechanism by which obesity induces onset and worsening of nephropathy are still unclear, and there is limited information on the development program of obesity-induced kidney injury in offspring of obese parents. The rapid increase in obesity in related cardiorenal and metabolic disease cannot be explained by change in genotype, but may result from environment factors in the interactions with genes. Maternal obesity is associated with a greater risk of hypertension in the offspring. It can also influence long-term energy balance. In fact, obesity is a preconceptual risk factor, especially in mothers that have BMI of over 30%. The obesity would have impact in the women, in the fetus, and also in the children. In the women, they have increased risk of miscarriage, gestational diabetes, in perinatal complication, and also low breastfeeding rates. And on the fetus, they have increased in risk of stillbirth, metabolic abnormalities, and also developmental abnormalities. And when the children, they would have increased risk of obesity and diabetes. Maternal obesity is widely recognized for its mediated complication in terms of pregnancy, such as gestational diabetes, as I mentioned before, preeclampsia, in the liver of large for gestational age infants. And here in this flow chart, you can see clearly that the maternal obesity normally is associated with increased gestational weight gain. They also have insulin resistance that will expose the fetus to altered glucose metabolism, increase in inflammatory cytokines, increase in circulating lipids. Also, they will have impaired placental transport in the epigenetic change that would lead to altered fetal placental growth. When the kids are born, they would have increase in the deposit, and also they have programmed to develop some metabolic disease such as diabetes and others, and also cardiovascular disease. When you think about how we can transmit the information from the parents to the offspring, there is a potential mechanism of transfer of this information from ancestral environment or physiological factors over generation. Here, as you can see, we have some triggers. For example, environment, that will be genotype or physiology. They would have effects on the father and also in the mother. And the father can transfer this information to offspring to the small no-code RNA, microRNA, and the transfer small RNA, and also through histone modification and DNA methylation. And also indirect effects via mother through the microbiome transfer. The mother also have effect of transfer this information to the offspring, not only through the DNA methylation as well, histone modification, but also include the postnatal care during the breastfeeding, and also directing the organelles such as mitochondria. Clinical and experimental evidence suggests a link between maternal obesity and cardiovascular disease in the offspring. Parental obesity and excessive gestational weight gain may contribute to increased obesity in the offspring, both in childhood and adulthood. In addition, adverse effects of poor fetal environment of the offspring may be carried to subsequent generations. Although has been considerable research on metabolic abnormality in the offspring of obese mothers, the long-term consequences of parental obesity on cardiorenal dysregulation in subsequent generations are still poorly understood. The mechanism by which obesity induces onset and worsening of hypertension and nephropathy are still unclear, and there is limited information about these complications. The link between parental obesity and long-term consequences of vascular and metabolic disease in the offspring was investigated in pups from male and female parents fed a high-fat diet and normal diet. As you can see here, the parents we call as F0 and the offspring as F1. We use high-fat diet to induce obesity. We use what we call Western diet, that we have 45% fat, 15% protein, and 41% carbohydrates. And the all-shall control, that's only 70% fat, 29% proteins, and 54% carbohydrates. When the offspring were born from those obese parents, we switched some to control diet. In some males and females, we kept the same diet the parents. In the control diet, the parents were fed the control diet and also the offspring. In some, we also fed a high-fat diet that we call F0 high-fat diet. Why you decide to use the CFIT7 mice for this experiment? These mice are resistant to developing kidney injury and have only mild, as you can see here, increase in blood pressure when fed a high-fat diet, even for 1 year, as you can see here. Similar to the mild hypertension and slowly developing kidney injury that occurs in humans, in obese humans. Using parental obesity, we designed an experiment to examine if intergenerational obesity leads to greater metabolic dysfunction compared to high-fat diet-induced obesity. Body weight was measured from 6 to 22 weeks of age in male control mice, as you can see here in black circle. We also have animals that the parents were fed a control diet, but we switched it to high-fat diet, that this group here in gray. Those animals born from obese parents that we kept on the same diet, we have here this group that is in the blue circle, and those that we switched to control diet, as you can see here in green. I hope that you can appreciate that these offspring from the obese parents that we kept on the high-fat diet, they are heavier than those fed a high-fat diet, but from parents fed a control diet. We did not see any major effect on body weight in the animals that we switched to control diet from obese parents. This increase in body weight was mainly due to increase in fat mass. As you can see here, F1 high-fat diet, they have large amount of fat, followed by F0 high-fat diet. Interestingly, we also found that at 22 weeks of age, the F1 normal diet that we call those, again, those are offspring that we switch to control diet, but they are from parents fed a high-fat diet. They have approximately 20% more fat compared to control animals. We also found increase in lean mass in those fat groups. This data suggests that inter-uterine exposure to high-fat diet robustly influence the fat storage. We then measure not only metabolic but also cardiovascular function in these animals at 22 weeks of age. In here, just to show you the phenotype at this age, that is showing that despite similar calorie intake in F1 high-fat diet, in F0 high-fat diet, the F1 high-fat diet, they have a large body weight compared to F0 high-fat diet. And the control in F1 normal diet, they have similar body weight in caloric intake. Offspring from parents-fed high-fat diet have impaired glucose handling even when fed a controlled diet, as you can see here, those animals that we switched to controlled diet, suggesting that in utero exposure to high-fat diet impaired glucose control. This response to glucose load is very expressed when you look at the area under curve. That's the in response to glucose load that we gave by gavage, and you measure at different time points at 15, 30, 60, 9, and 120 minutes after the gavage, and we look at and measure the area under curve. As you can see here, the fat animals, they have increased area under curve compared to control mice, even those that we switched to control diet. The increase in fat mass that we saw in those fat animals, the F0, EF, and F1 high-fat diet, were associated with a significant increase in plasma insulin levels, as you can see here, but only the F1 high-fat diet, they have higher plasma leptin levels, even compared to F0 high-fat diet. This is interesting because it's very close associated with a significant increase in fat mass in this group here compared to F0 high-fat diet. We further examined the liver fat content by Oredo, the immunohistochemistry, and also used the echo MRI. And we found that F1 high-fat diet mice, they have much more liver fat, even compared to F0 high-fat diet. And here, the Oredo is saying you can clearly see that these animals, the F1 high-fat diet, have much more lipid droplets compared to F0 high-fat diet. Interesting, we also found more fat droplets in the liver in the group fed control diets when compared to control animals from parents fed a control diet. Offspring from high-fat diet mothers also, they have reduced energy expenditure that we also saw in the animals that we fed high-fat diet after weaning, and they have a significant lower motor activity. This finding highlights the importance of energy balance in motor activity on body weight control. And I think most of you are asking what happened with the female mice. Do they have similar phenotype when compared to male controls? And the answer is yes. Yes, they do have. As you can see here, the F1 high-fat diet are larger than F0 high-fat diet. And there is no significant difference in the control and F1 normal diet. And again, the increase in body weight was mainly due to increase in fat mass. As you can see here, they have a significant higher amount of fat in the body compared to F0. And interesting that the F1 in F0 high-fat diet, they also have increased in lean mass. At 22 weeks of age, the time that we performed most of the measurement, they had a significant increase in body weight, even compared to F0 high-fat diet, despite have similar calorie intake. They also have impaired glucose handling, as you can see here by the area under curve, not only those that we fed a high-fat diet, but also in the offspring that we switched to control diet. Different than male, the F1 high-fat diet, they have higher plasma insulin levels compared to F0 high-fat diet. They also have a significant higher plasma leptin level compared to F0 high-fat diet. The ORDO and also the EcoMRI shows similar phenotype in these animals that F1 high-fat diet, as you can see here, they have much more lipid droplets compared to F0 high-fat diet. However, the F1 normal diet, we didn't see much lipid droplets, different that we saw in the male mice. They also have reduced energy expenditure, as you can see here, but there is no significant difference in motor activity. To summarize this first part of my talk, we found that intergenerational obesity leads to greater body weight gain in fat mass, not only in males but also in females when compared to F0 high-fat diet in control mice. In addition, male but not female offspring fed a normal diet from obese parents fed a high-fat diet have higher fat mass compared to control mice. In both males and females fed high-fat diet or normal diet have impaired glucose tolerance and more liver lipids accumulation compared to offspring from lean parents. And we saw an increase in plasma leptin levels in the male and females, although insulin level was higher in the female compared to F0 high-fat diet, but both they had more than animals from lean and kept in controlled diets. We also found that male and female offspring fed a high-fat diet from lean or obese parents have significantly lower energy expenditure compared to control mice. And also male but not female, from these parents, the obese parents, they have lower motor activity compared to control mice. Next, we examine whether intergenerational obesity leads to higher blood pressure and kidney injury compared to high-fat diet-induced obesity. Surprisingly, female offspring fed a high-fat diet or normal diet, they have similar blood pressure compared to F0 in control mice, and also similar heart rate. Potential mechanisms are sex hormone or aldosterone levels, and we will perform experiments to examine the role of these two hormones in female mice. However, for the males, we found that indeed offspring from obese parents fed a high-fat diet, they have higher blood pressure compared to F0 and also control mice. As you can see here, they have around 14 millimeters of mercury increase in blood pressure compared to control. And even the F1 normal diet, despite having normal body weight, they still have higher blood pressure compared to F0 in control mice. We did not see any change in heart rate. These data suggest that there is sex difference in blood pressure control in offspring mice from high-fat diet parents fed a high-fat or controlled diet. In addition, those animals that we measure blood pressure 24 hours per day by telemetry, and we look at the mean arterial pressure lability over time. In here, I hope that you can appreciate the control mice here in black. You can see, as we expect, they have higher blood pressure during nighttime compared to daytime because they are more active during nighttime than daytime. We didn't see much difference when the group that we fed a high-fat diet, but they were from parents fed a control diet. However, the F1 normal diet and also F1 high-fat diet, I hope that you can appreciate here, they have much more labile mean arterial pressure when compared to F0 in control mice. We also found that those animals, they have the mean arterial pressure frequency distribution shift to the right, as you can see here in blue, compared to F0 in control animals. Further, we also measure the spontaneous bodily reflex sensitivity in these animals when we found that only the F1 high-fat diet, they have significantly reduced spontaneous bodily reflex sensitivity compared to F0 control diet in even a F1 normal diet. Looking at the kidney function, we found that the increase in blood pressure in the F1 high-fat diet was associated with increased urinary albumin excretion, as you can see here. It was independent of body weight. You can see that F0, F1 high-fat diet, they have greater albumin excretion compared to F0. And we also look at the plasma creatinine levels, and I hope that you can appreciate those animals. Also, they have higher plasma creatinine level compared to F0 high-fat diet. This data suggests that these mice have altered kidney function. We also measure 24 hours glomerular filtration rate, the GFR, and again, we saw a significant reduction in the GFR in the F1 high-fat diet compared to F0 in control animals. We also found kidney injury, as you can appreciate here, by thickness of the Bowman's capsule, expansion of glomerular mesangial matrix, and tubular metaplasia, as you can see here when you compare to control mice. And we also found increase in fibrosis, as you can see here highlighted in blue compared to control animals. You also observed ultrastructural glomerular change, as you can observe here in the glomerular basement membrane. It's thicker than control mice. And also we found less endothelial capillary fenestration that you can see here close to the glomerular basement membrane compared to control mice. In addition, we also observed increase in mitophagy in proximal tubes in the high-fat diet animals. Obesity is also associated with increase in EI stress, in oxidative stress, and we further examined if these animals would have more EI stress, in oxidative stress. In fact, we found that shock, a common mark of EI stress, is significantly higher in the F1 high-fat diet compared to control. And we also found a greater H4HNE stain in the F1 high-fat diet, as you can see here in the kidney sample from F1 high-fat diet compared to F0 control or control mice. The question that we ask is, what are the potential mechanisms leading to effects of obesity-induced development program of cardiorenal function in these mice? In one potential mechanism mediate this effect of parental obesity-induced development program of cardiorenal function is through synergistic interaction of obesity, related metabolic disorder, and increased blood pressure that leads to excessive activation of purinergic receptor 7. Purinergic receptor 7, or P2X7, is a purinergic receptor for ATP, and its activation triggers calcium influx, cytosolic calcium overload, EI stress, inflammation. In addition, mitochondrial dysfunction is involved in P2X7 mediated cell death. Now my screen is frozen. We further investigate in those animals that do not have P2X7, that we call the P2X7 knockout mice, and we fed them a high-fat diet, similarly what we did with the CF7 black mice. And I hope that you can appreciate those animals that we with a high-fat diet from obese parents, they have higher body weight when compared to control mice. Not only males, but also the females. They also have increased in fat mass and also in lean mass. These animals also at 20 weeks of age, as you can see here, they have increased in body weight and also the calorie intake compared to control mice. However, despite having this increase in the fat mass, they do not have increase in blood pressure. As you can see here, the F1 high-fat diet P2X7 knockout mice, they have similar blood pressure compared to control mice, male and also the female mice. They also do not have any difference in heart rate. As you can see here, they have similar heart rate, not only male, but also female. We further ask if these animals will be protected against kidney injury compared to CF7 control if we increase blood pressure and we use the aortic coarctation between the renal arteries. In this technique, we would have one kidney that will be submitted to a normal or even reduced intrarenal pressure, and the kidney above the coarctation would have increased the intrarenal pressure in high blood pressure. We performed this experiment in 8 weeks after we measure blood pressure, and we compared to blood pressure measured before the coarctation. And we found a significant increase in blood pressure. It's around 22-25 millimeters of mercury, not only in the CF7 control, but also in the P2X7. And we will look at some markers of kidney injury in the albumin excretion. As you can see here, it was significantly increased in the CF7, but not much difference we found in the P2X7 knockout mice. We also look at the KIM1, this is the marker for kidney injury. And again, we didn't see major difference in the animals after the coarctation compared to baseline. However, the CF7, as you can see here, they have a significant increase in this marker of kidney injury. And also in gout, there's a marker of tubular injury, the same phenotype. We saw increase in the CF7, but not in the P2X7, showing that these animals indeed are protected against kidney injury when we have increase in blood pressure. To summarize the second part of my talk, we found that intergenerational obesity in male, but not in female mice, is associated with increase in blood pressure. And they also have increase in albumin excretion in plasma creatinine compared to control. They do have lower glomerular filtration rate and marked increase in renal morphological change, as I showed you, the thickness of the Bowman's capsule, expansion of glomerular matrix in renal fibrosis compared to control mice. The F1 high-fat diet mice also have more apoptosis, mitophagy, ER stress, and oxidative stress in the kidneys. And they also exhibit increased levels of markers of renal injury, renal inflammation, and also kidney dysfunction compared to F0 high-fat diet. And we use the P2X7 knockout mice. We found that this activation of this receptor, this purinergic receptor, may partially mediate the effects of obesity-induced development program of cardiorenal dysfunction. As I mentioned before, obesity is a major risk factor for cardiovascular and metabolic disease. And here in this map, you can see that areas, again, that have high prevalence of obesity. We also have high prevalence of diabetes. Not surprisingly, we have increase in hypertension. And also we have increase in the heart failure hospitalization, suggests that the obesity is also associated with cardiac dysfunction. And we use this model to examine if this international obesity leads to cardiac dysfunction when compared to high-fat diet-induced obesity, that's F0 mice. We use the Vivo 3100 to measure cardiac function, and we didn't see any systolic dysfunction in these mice. I hope that you can appreciate that all of animals, obese animals, the F0 and F1 high-fat diet, they have similar ejection fraction compared to control, and no major change or no change at all was observed in the F1 normal diet at 22 weeks of age. However, male offspring from obese parents, they exhibit increased markers of diastolic dysfunction. I hope that you can appreciate here. We measure the isovolumetric relaxation time in not only the obese but also the lean offspring from obese parents. They do have higher RVRT compared to F0 in control diets. And also we look at the E by IVRT ratio, and again we saw reduced in the F1 high-fat diet and F1 high normal diet. A prime also showed a decrease in these animals, and we saw only increase in the EA prime in the F1 high-fat diet. This data suggests that parental obesity contributes to impaired diastolic dysfunction in offspring. We also measure collagen deposition in these animals. As you can see, you have control F0 high-fat diet, F1 high-fat diet, and F1 normal diet. And I hope that you can appreciate it, that we have much more collagen deposition in the F1 high-fat diet and F1 normal diet compared to F0 in control mice. Interesting, we also found increase in lipid droplets in the heart, as you can see here in these slides. When we look at the EM pictures, we also found, as you can see here, the lipid droplets between the mitochondria and mitophagy in these animals and offspring, obese offspring from obese parents. Because of this increase in mitophagy, we decided to measure some marker of mitochondrial biogenesis. In one of them, it was the PGC1 alpha, but we didn't see major difference in these animals. As you can see, they have almost similar levels of PGC1 alpha compared to control, and there is no significant difference compared with F0 high-fat diet. We also look at markers of EI stress, SHOP that I mentioned before, for kidney, but we measure in the heart sample, and you can see that the SHOP is a significant higher the expression compared to F0 or control mice, and also we found the phosphorylate ERE1 alpha. We didn't see a difference in the interferon regulate factor. That's a marker of inflammation in the heart. As you can see here, F1 high-fat diet, F0 high-fat diet, they have a similar protein expression of the RF protein levels. However, we did not see any major effect in NF-kappa B in this samples from these offsprings. We further use exercise testing to assess the cardiovascular fitness, and we use a protocol that was developed by Petros when we increase the speed and also the incline with different time in each stage. And for the male animals, we did not see a difference in the performance, although we saw a significant reduction in the maximum velocity in F1 high-fat mice. They do also run less, as you can see here, compared to F0 high-fat diet, but we didn't find a major effect in the F1 normal diet. But the lactate level that we measure in compare before and after exercise, not only F1 high-fat diet, but also F1 normal diet, they had higher blood glucose lactate compared to F0 in control mice. And about the female, they also have no systolic dysfunction, as you can see here by ejection fraction, But we found a significant reduction in the E prime EA prime ratio, as you can see, the F1 high-fat diet and also the F1 normal diet. They do have increase in RVRT, and different than the males, they do have reduced in performance. As you can see here, the F1 high-fat diet and F1 normal diet, they do have reduced performance compared to F0 and also control mice. Then to summarize, I showed you that obese and lean offspring from obese parents have preserved ejection fraction, but exhibit signs of diastolic dysfunction compared to control mice. Those male offspring fed a high-fat diet from these obese parents have much more collagen deposition, EI stress, and inflammation in the heart compared to control mice. Female mice also showed reduced fitness, large increase in blood lactate level during an exercise test compared to control mice. In the female offspring, they do have preserved ejection fraction, but also have signs of diastolic dysfunction compared to control mice. And they also have reduced performance when we look at the response to exercise. These offspring fed a high-fat diet from obese parents have impaired diastolic function and reduced exercise tolerance with preserved ejection fraction. To conclude, we found that these offspring are more obese than those from obese parents and have greater metabolic dysfunction, including fat liver and impaired glucose tolerance. We also found that male but not female obese and lean offspring from obese parents have increased blood pressure, suggesting sex difference on blood pressure control in this model of obesity. The male offspring also have reduced GFR and increased blood creatinine levels associated with renal structural change in apoptosis. The parental obesity is also associated with diastolic dysfunction, increased cardiac EI stress and inflammation, and reduced exercise capacity in adult offspring. And the question is, what will be the next? We are trying to intervene, use diet, rich in the medium chain and short chain fatty acids to see if they would prevent this cardiometabolic alteration in these offspring. This, I would like to acknowledge all the team that has been working this project, include Dr. Hall, Dr. Alexandre Silva, Dr. Zhen Wang, Dr. Fabio Gavi, that helped a lot with the cardiac phenotype in these mice. So we still work on that, and this has been a pleasure to add more and more data and see the phenotype in these mice. We also have now in the lab Dr. Anna Moult that has been also helping us. We have Dr. Lee, Dr. Morton, and some students, undergraduate students from Brazil, Matheus Salgado, Gabriela Mega, Gisele Borges, that helped us to the quantification of the oradol and also the fat liver content and collagen deposition in the heart. And other members of the department, our technical support, and also our core that has been very helpful for us, in the funding support from NIH in our cover. And that is the end, and I'll be more than glad to help to answer any question. And I hope that you guys have learned a little bit about this intergenerational obesity and what the impact it would have in the offspring. Thank you very much.
Thank you, Jussara, for your wonderful speech and all your data. I can imagine that you have recent data and that you show it for us. So thank you so much for this as well. Thank you. So we have here some questions. So let's start with these questions from Anna. Is it possible that the implementation of an adequate diet and lifestyle in the gestational period of an obese woman helps to reduce the effects related to obesity in the offspring?
Yes, the question is most likely yes. We have a couple of data showing that intervention, especially in diet rich in fibers, for example, they would prevent some of the metabolic phenotypes in the offspring. As I mentioned at the end of my talk, we are also use the dietary intervention, the offspring, but we are also planning to intervene the mother to see if it would have impact in the offspring. And we are perform some experiments, use two different diets, the short chain fatty acid, the medium chain fatty acid, to see if you would prevent this effect. But most likely, if you intervene, for sure, not most likely, but for sure, if you intervene during the intra-uterine time, most likely you're gonna be prevent most of the metabolic phenotype in the offspring. But based on our data, it seems that the offspring, even if you put this offspring in the health diet, they still have some of the metabolic and also cardiovascular dysfunction. It's something that is you have to worry about it when you are planning to get pregnant.
Yeah. Yes, thank you. So the next one is about genes. In relation to the genetics of obesity, the genes responsible for the transcription of adipocytokines are the main involved, or could have other genetic components that, added to environment influence, could explain the perpetuation of obesity?
Yeah, this is always a good question, right? It's the gene or the environment. For sure, it's the combination of the two. They have some genes that you're gonna like trigger your or alter your, for example, your energy metabolism that would contribute to increase in the body weight and fat mass. But for sure, the combination of the two is gonna be play a major role in the obese phenotype. Lifestyle is something that we know that is very important. In our lifestyle, it has been modified over years, the decades, right? If you compare our lifestyle today compared to 10 years ago, it's much different. It's much more like sedentary kind of lifestyle than it used to be in the past.
Good. Thank you. Next one is about sex difference. So, are there results showing the proportion of transgenerational obesity related to the sex? If it is more frequent in male or female?
Yeah, this one thing that the offspring, for example, if you think about the body weight regulation, it seems that there is no much difference in male and females, at least in offspring born from obese mothers. Some they have increase in body weight, some they have low birth weight. But it's hard to tell because there are not many data showing the consequence of obese in the offspring, since that obese is a new alteration that we have in the human population. But based on the animal data, it seems that there is a sex difference. The males seems to be more prone than the females.
Thank you. And I think we have less questions, Jussara, so we are using all your time, so thank you. So the next question is from Giovanna. When we are scanning obese dogs or cats with the echo, we always have a hard time in order to have good images. Do you have the same problem with these obese mice?
Oh, you can answer this question, right? And if you ask the next speaker, he was also complaining a lot about it. But luckily, we have the Vivo 3100 that giving us a much better image. They have the high resolution image that help us a lot. For sure, it's a challenge. For example, in animals, if you compare the image that you are getting now with the 3100 with the one that we got back when we had the 770, it's completely different. But yes, we have a hard time sometimes to analyze the data because of the image that are not so clear.
Yes. Yes, good to know, because we suffer a lot here with obese dogs and cats. So yeah, so she's wondering about like the mice that are really smaller. So
Mhm.
yes, but we have Visual Sonics, right? You have
Yes,
Visual Sonics, so
yes,
how I
you're gonna
miss
have it.
Visual
Yes,
Sonics machine.
yes.
Okay, Jussara, so thank you so much. It was a pleasure to have you here, even remotely. So we hope that someday you can come and visit us here in Londrina State University. So in the name of our university, I would like to say thank you so much for your wonderful speech. Thank you.
Thank you, Fabio.
Okay, see you. Pessoal, então agora a gente vai dar um intervalinho e a gente já vai chamar o próximo palestrante aqui para a sala e a gente volta, é o mesmo link, tá? Então vocês podem manter conectados no mesmo link do YouTube que a gente volta aqui às 18 horas. Muito obrigado por enquanto. tarde a todos novamente. Então, agora eu vou virar a chavinha de novo, vou falar em inglês para apresentar o nosso próximo palestrante. So, our next speaker is Dr. Alexandre da Silva, also from University of Mississippi Medical Center. Dr. Alex da Silva started his career in UMMC in 2001 as an instructor in Physiology department. After that, he had the positions of assistant professor, and since 2018, he is a scientist in the Physiology and Biophysics department in UMMC. Dr. Alex's research has focused on understanding the mechanisms of central nervous system control of glucose homeostasis and cardiovascular function. Today, he will present to us the talk, Leptin, Cardiac Function, and Glucose Homeostasis. So, Dr. Alex, thank you so much for your presence here. It's a pleasure for us to have you here. As I always say here, one of examples that I would like to follow, one of the most intelligent people that I know, person that I know. So thank you so much for being here with us, and I wish you a good talk. Thank you.
Thank you, Fabio. You don't need to lie to please us. We miss you here, by the way.
Thank you.
It's getting cold, like when both of us were here in 2018. Remember, the end of 2017, beginning of 2018. It was a lot of fun. Okay, so hello everyone, and I'll share my screen. I think okay. And I'm gonna turn my cam off, so I think it may be better for you to just watch. Okay, so hopefully everybody can hear me. So today I'm gonna talk still about leptin. Dr. Hall, I think, glanced a little bit about the effects of leptin on blood pressure regulation and obesity-induced hypertension yesterday. But today I'm going to talk more on some positive aspects of leptin, as it may be protective against cardiovascular dysfunction after myocardial infarction, and how it can help regulate glucose homeostasis even in the absence of insulin. So the talks that I have planned today, if we have time, I may go a little over, so I may shorten it a little bit, but is to begin talking about this powerful central nervous system mediated cardiometabolic effects of leptin. For example, I'm going to show you some very compelling evidence that leptin, via its effect on the central nervous system, can normalize glucose levels in type 1 diabetic animals and improve heart function after myocardial infarction, preventing against progressive heart failure. And I also am going to show you some data that shows that the brain melanocortin system, particularly melanocortin-4 receptors, is a key component of these beneficial effects. And if we have time, the last 2-3 slides, I'm going to show some of our recent data with 4D echocardiography, which may better estimate systolic function after myocardial infarction, which may be underestimated when we use the standard B mode, for example. So yesterday, I think Dr. Hall described to you the leptin and its effects. So leptin's major role, or for what it's best known to act in the body, is a major regulator of appetite and body weight. So lack of leptin, you have the OB and the DB mice, which have leptin, defective leptin receptors. So you have massive early onset obesity. And this has also been translated in some rare forms of early onset obesity in humans that don't express leptin or have a defective leptin receptors. But leptin, in fact, has a variety of other important effects in the body. Yesterday, Dr. Hall mentioned some of its effects on blood pressure regulation. And today I'm going to talk about our major interest, which is the effects of leptin on glucose homeostasis, which will be the first part of my talk today, and on cardiovascular function, particularly cardiac function, on the remaining half. So since its discovery in 1994 by Jeff Friedman, his group also showed very soon afterwards that acute infusion of leptin for a period of 3 to 6 hours in rodents, in mice in that case, improve glucose turnover in the whole body. So what it means is that in the presence of high circulating leptin levels in lean animals, then the body requires more glucose to maintain euglycemia. So the uptake of glucose by all the tissues was improved. And this can be highlighted here, where you can see that glucose uptake in most of the tissues is increased in the animals that were acutely infused with leptin. This has also been shown in humans. So here are data in lipodystrophic patients with lack the capability to produce normal amounts of leptin. As you can see, their plasma leptin levels are very small. And despite that, they are lean. As you can see here, their BMI is actually below the lean controls, but they have severe insulin resistance, hyperglycemia, and hyperinsulinemia. And when these individuals were treated with leptin for a period of 12 months to normalize their leptin levels, as you can see here in the treated group, it markedly improved their insulin sensitivity and their glucose values. In this other study, although this is only one patient, but it was an adolescent with acquired lipodystrophy due to type 1 diabetes that was being treated heavily with high dose of insulin, as you can see on the top here. Then the investigators began to treat this individual with leptin on daily injections for 14 months. And you can see that daily leptin injection in this individual markedly reduced the requirements for the daily dose of insulin, while at the same time markedly improving the glucose profile and also reducing triglyceride levels in this individual, showing that leptin may have an impact on glucose homeostasis in humans as well as in other animals. So at that time, when I was interested in this role of leptin on glucose regulation, most of the current paradigm of that was that leptin improves insulin sensitivity, and that would then help regulate glucose homeostasis. But we thought that perhaps leptin may also regulate glucose homeostasis via insulin-independent effect. So to test this hypothesis, we induced type 1 diabetes in rats. So these are Sprague Dawley rats. So we inject streptozotocin to destroy the pancreatic beta cells that produce insulin. And the rats become severely diabetic, as you can see in this group here, which is not treated and has very severe diabetes. But then we began to treat the animals after day 5 post-injection of STZ with leptin intravenously. And it is the group with the black circles. And as you can see, with time and leptin treatment, blood glucose levels return almost all the way back to baseline values. However, to our surprise, when we gave leptin directly into the brain at a very low dose that did not spill over into the systemic circulation, which is the group with the white circles, then glucose levels fell even more rapidly and remained in the normal range while the animals were being treated with leptin, just suggesting to us that leptin has a powerful anti-diabetic effect that is independent of any effect that it may have on insulin sensitivity, and that this effect is likely mediated by the central nervous system actions of leptin and not by its peripheral actions. We then repeated these experiments in animals that were also diabetic, were not treated with leptin, but were pair-fed, because leptin does reduce food intake, and one of the questions we had was if this improvement in glucose homeostasis in diabetic rats was due to the fact that leptin was just making them eat much less. But as you can see in the groups represented by the dark triangles, the black triangles, they ate the same amount of food compared to the animals treated with leptin, but their glucose levels was in the 300, 350 milligrams per deciliter compared to the leptin-treated groups, which was completely normal, around 80 to 90 milligrams per deciliter. And here is just to highlight how powerful leptin is. So these are urine volume and water intake during baseline, when the animals are normal, before the injections of XTZ. Then we induce diabetes, so it's very severe diabetes. You can see that the animals, these are rats that are excreting the same volume of urine as their body weight. So it would be the equivalent of a human urinating 70 liters a day, for example. But with leptin treatment, either intravenously or directly into the brain, we can completely normalize urine volume and water intake. Actually, you cannot distinguish a normal animal from a diabetic animal that's been treated with leptin. To give an example of how powerful leptin is in this type 1 diabetic model. Then we began to study the potential mechanism by which leptin mediates these anti-diabetic effects. And one obvious candidate is the brain melanocortin system that Dr. Hall also mentioned in his talk yesterday. So he showed yesterday that leptin activates POMC neurons in the arcuate nucleus and perhaps in the nucleus of the tractus solitarius as well. That then leads to the release of alpha melanocyte stimulating hormone, which would then activate melanocortin 4 receptors. For example, when we block, he showed you this yesterday, when we block these receptors, then leptin loses its ability to reduce food intake and to increase sympathetic activity, blood pressure, and heart rate. And then we ask the question, is this system also involved in the anti-diabetic effects of leptin? So one of the first things we did was to repeat our experiments with leptin. So what we've done is repeat. So we have regular animals, we induce diabetes, they become diabetic. And then here we treat them with leptin. You can see that completely normalize their blood glucose levels. But in another group, a similar group, we pre-treated them with SHU9119, which is a melanocortin 4 receptor antagonist. And then we began giving leptin. However, in this group, as you can see, leptin had no effect to reduce blood glucose levels. In a subsequent study, a couple years later, we repeated this protocol, but we decided to test in a different way, because it could be possible that this MC4 antagonist could somehow have some offset effect that was, making the animal sick or altering the ability of leptin to bind to its receptor. Who knows what that drug could do. So what we did, we used melanocortin 4 deficient rats. So we use a genetic approach. We induce diabetes, as you can see here. So the white bars are wild type controls. The black bars are melanocortin 4 deficient rats. And then we treat them with leptin ICV, intracerebroventricularly. And as you can see in the control animals, blood glucose was markedly reduced, which was not observed in the MC4 deficient rats. Again, comproving our previous study showing that the melanocortin 4 receptors are required for leptin to exert its anti-diabetic effects. We went a step further, and although this is preliminary and unpublished observation yet, and we used mice with streptozotocin-induced type 1 diabetes as well. And these mice have no functional melanocortin 4 receptors anywhere in the body except in neurons of the paraventricular nucleus, which we created using a Cre-Lox recombination of SIM1 Cre with LOX-TB MC4 mice. And when we rescued melanocortin 4 receptor in this paraventricular neurons, nuclear neurons, then the animals began to respond, as you can see. So this is they have basically a similar response, anti-diabetic response to leptin as a wild-type mouse. So it appears that melanocortin-4 receptors in paraventricular nucleoneurons are very important in mediating these anti-diabetic effects of leptin. However, the story gets a little bit more complicated. So we've shown that if you don't have functional melanocortin-4 receptors, then leptin will not work. So it will not have its anti-diabetic effect. So then we thought, okay, so if we just activate melanocortin-4 receptors, then perhaps we can mimic what we see with leptin. But the matter of fact is that that was not the case. So we used the melanocortin-4 receptor agonist called MT2, and we used several doses, but it failed to normalize glucose levels in diabetic animals. So that suggested to us that leptin does activate the brain melanocortin system, but it may also suppress another system in the brain that opposes the actions of the brain melanocortin system. So the combination of these actions then result in this powerful anti-diabetic effect of leptin. And in fact, another group working on the same area published later on that if you rescue leptin receptors in POMC neurons, which are needed for activation of the melanocortin-4 receptors, but you also activate leptin receptors in glutamatergic neurons combined, then you can rescue the totality of leptin's anti-diabetic effects. So this may be a little bit of a complicated figure, but what the authors were trying to show here is that using also this type 1 diabetic model, if you don't do anything with the animals, in about a month after diabetes, then about 50% of them die if they are not treated. In the animals that they rescue, well, in the animals that they give leptin to regular animals, then 90% of them survive. And in the animals that do not have leptin receptors, except leptin receptors in in GABAergic and POMC neurons, then they can also keep about 90% of them alive. And here, as you can see in the grayish circles, their blood glucose levels also go back all the way to normal values. So, as Dr. Hall showed you yesterday, and I briefly showed today, leptin activates the POMC neurons, which are glutamatergic neurons. They are, how do you say, they also express glutamate. But leptin, in addition to activate POMC neurons, it also inhibits AGRP NPY neurons, which actually are located besides or in this almost in the same area of POMC neurons in the arcuate nucleus. And these neurons are GABAergic. And the function of the NPY and AGRP is to antagonize the action of the brain melanocortin system, either by inhibiting the depolarization of POMC neurons or the polarization of other second-order neurons that also express MC4. So one of the first things we tried to do was to investigate the role of NPY or neuropeptide Y as a compensatory factor to offset the effects of melanocortin-4 receptor activation. So what we've done is we purchased NPY deficient mice, we induced diabetes, and we gave the MC4 activator MT2. But what we found is that in the animals that have normal NPY, as well as in animals that lack neuropeptide Y, chronic activation of the melanocortin-4 receptor did not reduce food intake or reduce glucose levels in these diabetic mice. So there is also a possibility that the AGRP, which is a GUT-related peptide, which is also an endogenous antagonists of melanocortin-4 receptors may play a role. It's something that we are also investigating now, or that may be some other factor that offsets the impact of melanocortin-4 receptor activation with regards to the anti-diabetic effects of leptin. And it's an area that we are still working on to understand better this interaction of the leptin, POMC neurons, AGRP and EPY neurons, and the activation of melanocortin-4 receptors to translate into marked improvement in glucose regulation in diabetes, even in the absence of normal insulin levels. But then now leaving the brain a little bit and focusing more on the periphery, we also were curious of how this powerful brain-mediated anti-diabetic effect of leptin can be transmitted to peripheral tissues. So we know that to maintain your glycemia, you need a balance of glucose input and glucose utilization, which mainly is what you eat and what the liver produces, by what is stored and what is metabolized by the tissues. And another group friend of us that also working in this area showed that in diabetes, in type 1 diabetes, you have a marked increase in gluconeogenesis by the liver, and leptin completely normalizes this, the increase in liver glucose production, while at the same time increasing glucose uptake by peripheral tissues. And in fact, another group, so these are very old studies, somewhat old, 20-year-old study, showing that if you acutely inject leptin in the brain, you increase glucose uptake in several tissues, and that can be blocked if you denervate the tissue. And in another group showed that microinjection of leptin in the medial basal hypothalamus also increase glucose uptake, as you can see here in the heart and the soleus muscle, and that can be blocked by propranolol, which I think you all know it's a beta 1 and beta 2 adrenergic receptor antagonist. So all these acute studies pointed out that the autonomic nervous system could play a major role in mediating the effects of leptin on glucose regulation by peripheral tissues. So what we've done is then to test if the chronic anti-diabetic effects of leptin that we observed was also mediated by the sympathetic nervous system, for example. So what we've done is to repeat the diabetic study in rats, but this time we gave we gave propranolol, and we gave also an alpha-1 antagonist, terazosin, to these animals to test if there was a role for the sympathetic nervous system. But as you can see, adrenergic receptor blockade did not attenuate the anti-diabetic effects of leptin. We then also focus on the impact of leptin on liver glucose production. So previous studies have also shown that leptin injecting into the arcuate nucleus reduces liver glucose production, and that can be blocked by vagotomy. So what the author suggests is that leptin increases parasympathetic activity to the liver, leading to a reduced production of glucose. So we attack this question in two ways. So I'll show you the one on the right first. So Jussara and I, Dr. Jussara do Carmo and I, we induced diabetes in rats, and we performed hepatic denervation, vagal denervation. And what we saw is that even in the denervated rats, if you give them leptin ICV for 7 days, you completely normalize their plasma glucose levels. Then in other rats, we also use this protocol. We induce diabetes, they become diabetic, then we treat them with leptin, and they become normal glycemic. And here we use hexametonium. So hexametonium was given, to these animals are treated with saline, and these animals are treated with leptin. And we gave hexametonium to both groups. Hexametonium is a ganglionic blocker, so we are blocking both sympathetic and parasympathetic inputs to peripheral tissues, including the liver. And as you can see, that even in the presence of hexametonium, leptin continued to maintain euglycemia in insulin-deficient diabetic rats. So then we thought, well, if it's not the autonomic nervous system, then it may be that leptin triggers the production of a neurohumoral factor that acts in the tissue to kind of mimic what we see with insulin. And so we cultivated skeletal muscle cells, immortalized skeletal muscle cells, and we exposed them to plasma that contained 10% of, we exposed them to media, to culture media that contained 10% plasma from diabetic rats that received leptin ICV. And our initial studies showed that glucose consumption by these cells was markedly improved compared to control cells that had the regular media with plasma from diabetic rats that were not treated. And this also happens in the presence of insulin in the media. So this first in vitro evidence suggests that there could be a circulating factor triggered by leptin that would have a powerful anti-diabetic effect. Then Jussara and I used parabiotic experiments. These are some, these are actually experiments that first evidenced the existence of leptin in the 60s, if I'm not mistaken. So she joined Lewis rats when they were very young, at 4 to 5 weeks of age, so they have cross circulation. All of the animals were made diabetic, and they had ICV cannulas, but only one animal, for example, of this pair, one animal was treated with leptin, one animal was treated with leptin, and let's say his brother here was treated with vehicle, and the other pair was only treated with vehicle. And using this protocol, what we found was the animals that are only treated with vehicle, they remain markedly diabetic. The animal that receives leptin in the brain, so his glucose falls to normal values. And the animal that is attached to him, which in this case would be this animal here, shows somewhat an intermediate phenotype. Actually, glucose levels here is about 22 to 27% lower than the vehicle-treated animals. And when we measured glucose production by the liver, then we can see that in diabetes, there's a marked increase in liver glucose production compared to control non-diabetic rats. Leptin completely normalizes it in the animal receiving leptin, and it reduces by about 25% in the animal that did not receive leptin, but that shared the circulation with the animal that was treated with leptin. So this result also suggested to us that there may be a circulating factor responsible for this anti-diabetic effects of leptin. And we then tested if there is a circulating factor, would it be coming from the pituitary gland? So we ordered hypophysectomized rats from Taconic here at United States, and then we also made them diabetic, and we gave them leptin like we've done in all the previous studies. But leptin still was able to normalize their blood glucose levels despite lack of pituitary hormones. So to summarize what I've shown you so far on these anti-diabetic effects of leptin, we know that it's mediated by the brain. We know that activation of the POMC MC4 axis is important. It is not attenuated by adrenergic receptor blockade or hepatic vagotomy. It's also not prevented by ganglionic blockade. I also showed you it's not due primarily to decrease in food intake by leptin, and it cannot be prevented by hypophysectomy. So we also shown, but this is unpublished, that plasma from rats given ICV leptin increases glucose utilization in in cultured cells and reduces hepatic gluconeogenesis in parabiotic rats. However, unfortunately, the identity of this factor is still unclear, and we are having a hard time because, we are physiologists and not biochemists to identify this factor yet. But we'll continue to work on it, and hopefully we find something, in the next few years. Hopefully, we'll see. But while conducting these studies, we also noticed that this model of type 1 diabetes is not only associated with high severe hyperglycemia, but they also have, this model also exhibits some cardiovascular changes. And one of that is very clear is bradycardia. Okay, and one of the mechanisms that triggers this bradycardia is also a reduction in intrinsic heart rate. As you can see here, this is in rats. So this is intrinsic heart rate in Sprague-Dawley rats at baseline, and this is about 7 days after streptozotocin injection. The difference is about 50 beats per minute. And the intrinsic heart rate is the heart rate in the absence of sympathetic or parasympathetic inputs to the heart. And we noticed that with leptin ICV treatment, the intrinsic heart rate returned all the way back to normal. So we thought that leptin may be improving the metabolism of these pacemaker cells in the heart. And that, plus the fact that leptin improves fatty acid and glucose metabolism in several tissues, including the heart, led us to speculate that perhaps the leptin and the brain melanocortin system could also be important in protecting the heart in situations of metabolic stress, such as occur in heart rate induced by a myocardial infarction. So this is actually a very important topic because heart failure, especially congestive heart failure, is a very serious issue for which there is basically no effective therapy. Here in the United States, I guess in Brazil as well, 1 in every 4 deaths is due to heart disease, and a significant portion of that derives from heart attack and myocardial infarction. So development of new and more effective therapies are, urgently needed. So actually, Fabio began these studies with me in Brazil, and then when he came here, he obtained several of the data that I'm going to show you today, and I continue on this project. So some of the data that I'll show you today were obtained by Fabio, and some are obtained by myself and Anna and others in the lab. So what we've done here is we instrumented the Sprague-Dawley rats with ICV cannulas. Then after they recover for a week or so, and we do normal control measurements, including echocardiographic evaluation using the Vivo 3100 that Jussara mentioned in her talk. Then we tie the left descending coronary artery. So this is permanent ligation. It's not ischemia reperfusion. And then we follow the animals for 4 weeks. And in some of the animals, we treated with saline, which is our vehicle, or leptin in the brain using the same dose that we used in previous studies examining the anti-diabetic effects of leptin. And we perform weekly echocardiographic measurements. And as you can see, the animals after the myocardial infarction, they develop progressive heart failure, as you can see by a reduction in ejection fraction, as well as in cardiac output. But the animals that received leptin ICV showed a much better recovery and actually have a much better, ejection fraction and a completely normal cardiac output by weeks 3 and 4 of treatment. What we also saw is that leptin ICV treatment prevented the enlargement of the left atrium, which is an index of cardiac congestion, as you can see here on the right. So the animals not treated, the left atrium begins to increase, which doesn't happen in the leptin treated animals. And we also saw an increase or a restoration of radial strain of the left ventricle compared to vehicle-treated animals. And this is actually, even when you take the area 6 and 1, which basically comprise the anterior wall, which is the area that's most affected by the infarct. So this suggested that the effect that leptin is having in the heart is not because it prevents the heart failure, but actually it improves the function of the cardiomyocytes in the areas that are functioning well and were not affected by the infarct. I think this perhaps better shown here. These are videos I think obtained by Fabio as well. So this is the same animal before the infarction. This is week 1 of the infarction. You can see there's a huge infarct here. And this is week 3 of treatment. As you can see, the infarct area is still much akinetic, but the other areas away from the infarct move much better. And this is a short axis view before, on week 1, and on week 3. And we also observed that there was less of an increase on heart weight in the leptin-treated animals, as well as in lung weight. And the animals treated with leptin also continue to perform equally on a treadmill compared to the animals that received saline. So as you can see, this is before the MI, the myocardial infarction, and this is week 4 of the MI. So the animals, as expected, they can run much less, which did not occur in animals that are treated with leptin. So then again, we began to think, is the brain melanocortin-4 receptor involved? So how did you test the involvement of the brain melanocortin system? So basically it's the same line of thought that I showed you before for our earlier experiments. So one thing that we've done is we repeated the leptin infusion, but in this case in melanocortin-4 deficient rats, or in Sprague-Dawley rats, we gave the melanocortin-4 receptor agonist, MT2. So first, what we found is that in melanocortin-4 deficient rats, leptin failed to improve cardiac function. So suggesting that functional melanocortin-4 receptors are needed for leptin to have this marked effect to improve cardiac function post myocardial infarction. But contrary to what we've seen with the anti-diabetic effects of leptin, if we only activate melanocortin-4 receptors with MT2, we can recapitulate what we've observed with leptin. So here on the gray bars is melanocortin-4 receptor activation with MT2, and you can see that these animals recovered much better from the infarct, as well as the animals that were treated with leptin. So this here showing cardiac output, ejection fraction. It also attenuated, actually completely prevented the enlargement of the left atrium, and also markedly increased left ventricle radial strain. So this shows to us that then melanocortin-4 receptor activation is completely capable of ameliorating cardiac function like leptin post-myocardial infarction. And now what we don't know yet is the mechanism is involved. And so one of our earlier data that gives some hint of what may be involved in the heart at least is an improvement in mitochondrial function and mitochondrial number. So this suspicion comes from the fact that at 2 weeks of treatment, we find an increase in phospho-AMPK and PGC1 alpha, which are important regulators of cellular metabolism, as well as mitochondrial biogenesis. In fact, we see using electron microscopy that there appears to be much more mitochondria in the myocardium of leptin-treated animals, and the same is true for MT2-treated animals compared with vehicle. And Naomoto and I confirmed this data analyzing mitochondrial DNA. So there's much more expression of mitochondrial DNA here at COX-3 and ND4 in animals treated with leptin compared to with vehicle. And using cardiac fibers, we measured oxygen consumption by mitochondria. So this is a direct way of evaluating mitochondrial function. And what we found is that ATP-linked respiration, which can be used as an index of mitochondrial efficiency, is much better in leptin-treated animals, as well as in animals receiving the melanocortin-4 receptor activator MT2, compared with vehicle. And using isolated perfused hearts with the help of another professor here in another department, Romain Harmency, we observed that ICV leptin or MT2 infusion markedly increased glucose oxidation by the heart, as well as fatty acid oxidation. So what we see here is that there are more mitochondria, the mitochondria is more efficient, and the heart can metabolize better glucose and fatty acids. Then we isolated cardiomyocytes and we measured sarcomere shortening directly using ionics system here in our laboratory. And what we can see is that both sarcomere shortening as well as shortening velocity were markedly improved in leptin-treated, in MT2-treated animals. So here's a representative tracing of cardiomyocyte sarcomere length. So you can see that the shortening, which means how the sarcomeres are contracting, is much faster and much more profound in treated animals compared with saline. So what we believe is happening is that leptin acts in the brain, activates the melanocortin system, particularly melanocortin-4 receptors, which we also hypothesized are important in certain specific nuclei, which we are currently investigating. And this then leads to improved mitochondrial biogenesis, increased substrate utilization and energy production in the non-infarcted area, which then leads to improved cardiomyocyte contractility in this non-infarcted area, leading ultimately to a preserved cardiac function in heart failure. And now in the first couple minutes here, just to show you some of our recent data with 4D echocardiography. So all the data that I showed you so far is mostly B mode, where we use left ventricular tracing to evaluate systolic function. But when we do 4D, then we have, how do I say, not only the dimension, but also the time, so we can basically see the heart contracting. And in theory, you would expect to have a more, how would I say, a closer to true cardiac function using 4D. And what we've found so far, but these are data from only 4 or 5 rats, is that in animals with normal hearts, which means that the shape of the heart is normal, we have not found a significant difference using B mode or 4D. But in the hearts that have pronounced MI, so it becomes misshapen, then the 4D may be overestimating the decrease in cardiac function compared to 4D. So with 4D, we get better ejection fractions, cardiac function, and reduced end diastolic volume. And I think, although this needs to be confirmed, that this may be expected, because in a normal heart, as you can see, this is our 4D echocardiography video, then the heart changes shape from top to bottom and from left to right, as you can see, and it squeezes the blood out, as you can see here. But in an animal with MI, usually only the anterior wall is damaged. Then when you use the B mode, you can see that there's basically no movement from top to bottom, but from left to right, you see the heart continues to move basically normally. So this may be underestimated by using a 2D mode, like a B mode. And this can actually be captured when we use the 4D. And now to finish and trying to actually join both portions of my talk today. So what I think I've shown you today is that leptin, via activation of its receptors in POMC neurons, lead to activation of melanocortin 4 receptors in the paraventricular nucleus, and also inhibits the activation of GABAergic neurons, although we don't know the identity of these neurons yet. And these neurons, when they are not inhibited, can block the actions of melanocortin 4 receptors. But then what leptin would do is, in its chronic effects, would lead to the release of a neurohumoral factor that increases peripheral glucose uptake, while at the same time reducing liver glucose production, then leading to improved glucose homeostasis in long term, while previous studies have shown that leptin, perhaps also through activation of the POMC and MC4 receptor axis, then acutely would raise parasympathetic activity and sympathetic activity to peripheral tissues, leading to reduced liver glucose production, increased peripheral glucose uptake, then causing a short-term, usually minutes to a few hours, improvement in glucose homeostasis. We are still not sure how leptin improves cardiac function in heart failure, but it may be through an action of this neurohumoral factor that may improve the heart's capability of metabolize glucose and fatty acids, and may also improve mitochondrial function, or may also involve the autonomic nervous system, perhaps the sympathetic activity to the heart, perhaps activation of the cholinergic anti-inflammatory pathway, or perhaps a combination of modulation of autonomic activity as well as a direct action via a neurohumoral factor. So this is where we stand now. So we still have many unanswered questions regarding this project. And I'd like to finish by also acknowledging the people that worked in this project, Dr. Hall for his support and his mentorship, Fabio, who was very instrumental in getting this project started, Dr. Jussara, Donna Anamoto, Romain, who helped me run the perfused heart preparation, his wife, who ran some PCRs for us, and all the folks in the lab, people from Barão de Mauá, Ribeirão Preto, who came here for a couple months each time and worked hard in this project. Actually, I have them twice here. My bad. And for our financial support, and I'll be glad to answer any questions. I hope you guys have enjoyed and are not been too tired following 2 long talks today. Thank you.
Thank you, Alex. Can you just turn on your camera? Yes, please. Thank you. Thank you so much. We have here some questions. So thank you for your wonderful speech. I was dreaming with your 4D images, so I couldn't work with that one when I was there. So we have here one question from Professor Marley from Physiology Department here from Londrina State University. So in your studies, we observed an increase in oxidative stress in the RVLM area of obese rats associated with hypertension. Could this effect be due to leptin resistance in these animals? And also we have a part 2. Any indication that it is related to glutamate receptors?
That's a good question. Well, Dr. Ali Mark mainly have suggested that there is this selective leptin resistance where the effects of leptin to reduce appetite and perhaps to improve glucose homeostasis are impaired in obesity, but leptin's effects to increase sympathetic activity and then trigger hypertension are preserved. And I think that that may happen in humans as well as in rodents. So if that is the case, I think, yes, that maybe this increased stress that you see in RVLM neurons may be due to leptin. Although here, well, I have never actually investigated how leptin regulates ER stress as well as ROS production in neurons, but some have shown that leptin may increase ROS production, so it may be an important mechanism as well. If it's related to glutamatergic neurons, it's possible. I know there are PUMC neurons also in the brainstem. They are relatively less studied compared to the PUMC neurons in the arcuate nucleus. And I also think they are glutamatergic. So I don't know if how leptin regulates PUMC neurons in the brainstem, or if leptin would directly activate these neurons in the RVLM, or if it could be through other neuron that's activated by leptin, or the melanocortin system that then feeds back or feeds forward to the RVLM neurons and then triggers ROS generation and alters the regulation of those neurons. Ultimately, I think leading to sympathetic activity. So I don't know, but that is, I think that may be a possibility.
Okay, perfect. Thank you. So the next question is from Gabriela. Gabriela is our undergraduate student here from Veterinary College in London State University. If leptin can increase sympathetic activation, hypertension could be a side effect of its use in patients?
Yes, it could. Actually, it's been such a long time, but the reason I started studying the effects of leptin in diabetic animals was because I wanted to see its side effect, actually. But then we got detracted and we began to focus more on its anti-diabetic effects per se. So yes, that leptin could have this side effect. In the studies where they've replaced leptin in patients, some of which I showed in the introduction of my talk today, I don't recall them showing blood pressure data. So I actually don't know. I don't think they actually showed any blood pressure data. But there as well, they were just bringing leptin back to what would be normal values, because those humans had no leptin almost. So I think the effect of leptin in that case on blood pressure regulation would not have been much detrimental. But in other cases, like when you are raising leptin levels a little bit more pharmacologically, yes, there could be a side effect. That's why one of the good things of that Fabio and I, what we've been observing, is that if we activate the brain melanocortin system, we can probably bypass the need for leptin. And that is good in two ways. One, because, as Professor Marley mentioned, there may be leptin resistance. We also don't know if there is leptin resistance for the anti-diabetic or the cardiac beneficial effects of leptin. So, but they may develop in obesity. We don't know. But if we can bypass that by activating melanocortin-4 receptors, then we avoid the side effects of leptin and we avoid the leptin resistance. And there is a new melanocortin-4 receptor agonist that I'm beginning to try now, called setmelanotide, who have been approved by FDA recently to be used in genetic forms of obesity. And the authors of that study, they know of our studies that MC4 activation increases blood pressure, and they did measure blood pressure as a potential side effect on those patients. And this particular compound appears to have very little side effect regarding blood pressure. So it's a way to circumvent this side effect of leptin.
Perfect. Thank you so much.
I apologize for the long response, but I want to give more context to it. Sorry.
No, you're good. You're fine. Thank you. So the next question is from Patrick. In patients with insulin resistance, can we see leptin resistance as well?
Well, I think most patients with insulin resistance, they have some metabolic disturbances, I think. I mean, the metabolic disturbance usually leads to insulin resistance. So although they may not be severely obese, like we see here in US, like in Brazil, people are much less obese. But I think visceral obesity is the major culprit here. So even though an individual may not look very fat, may be overweight, he has metabolic disbalance, and that will induce insulin resistance. And I think in those individuals, there may also be leptin resistance. That's what I would bet, that they would have some degree of leptin resistance. So one of the things that we've talked about that is in obese individuals who have insulin resistance and have high leptin levels, does this prove that they are leptin resistant? Or if we were to block leptin levels on them, although leptin antagonists are incredibly expensive, it's very difficult to do those studies. But if we were to reduce leptin levels, would they be worse? You see what I mean? So maybe leptin is still trying to, to make them less worse. But we don't know because it's difficult to test that hypothesis. But if I had to bet, they are leptin resistant as well.
Okay, very good. And I think now we have the last question from Ana Paula. Knowing that obese people have more leptin, and you showed us good effects of leptin in heart function, do you think that leptin is enrolled in the paradox of obesity?
It may be, yes. We talked some about this obese paradox as well, that although obesity is one of the leading causes of several diseases, including myocardial infarction, because of hypertension, diabetes, etc. But obese individuals appear to do better after they have myocardial infarction, right? So I think it's a possibility, that despite the obesity and all the problems, that the ones that do survive may do better. Also difficult to test, right? The best experiment would be to block leptin on them and compare, right? Or in an animal. But obesity in animals is very different, especially in rodents, is very different than in dogs and in humans. Usually rodents don't develop severe hypertension, and it's different. So it makes it a little more difficult to investigate the mechanisms.
Yes. So thank you so much, Alex. Thank you for all your answers. Thank you for your lecture. Excellent talk. Thank you so much for your time with us. I really miss work with you all, but it was a very nice time yesterday and today. So thank you so much for this moment with us. In the name of Londrina State University, I say thank you. And it was a great time for us here. Thank you so much.
And if other students or other professors have any question, they can contact me personally, and email me. You have my contact information. You can pass to them, and I'll be glad to answer and the best I can to them.
Very good. Thank you so much, Alex. Thank you. Pessoal, então agora a gente encerra, os nossos 2 dias de palestras. Foi um prazer para mim poder intermediar essa conexão, UEL e UMMC. Então, eu gostaria de agradecer inicialmente à Universidade Estadual de Londrina, à Pró-Reitoria de Pesquisa e Pós-Graduação. Também gostaria de agradecer ao programa, né, de pós-graduação do Ciência Animal, em nome do professor Dr. João Luiz. Gostaria de agradecer também as meninas da organização desses webinars, que elas foram, assim, excelentes e impecáveis. Não vou falar o nome de todas aqui, mas saibam que eu estou estendendo meu agradecimento a todos vocês da organização. E eu também gostaria de agradecer a todos vocês que estiveram presentes ontem e hoje, né, espero que os temas tenham, né, agradado a todos. Eu fiquei extremamente feliz, né, com essas apresentações de ontem e hoje. Então, foi um prazer para mim. Continuem se cuidando e lembrem-se que vai passar, tá? Então, fiquem todos com Deus e até uma próxima. Tchau, tchau, pessoal. Muito obrigado a todos.